Welcome to the subunit on the PCR-based molecular markers. This subunit is part of the Unit 14.1 Plant Genotyping Genetic Markers within the module M14 Molecular Markers and Genetic Mapping. By the end of this subunit, you will be able to compare the different types of markers, analyze the pros and cons of every marker, and illustrate the advantages of PCR-based molecular markers. In the previous subunit, first generation of molecular markers, we have reviewed the first types of molecular markers based on DNA, RAPDs and VNTRs that are amplified by restriction enzymes and hybridization. A new revolution in the genotyping came with the introduction of the PCR. The PCR polymerase chain reaction allows the amplification of selected regions of DNA, even with a small quantity of it. Thus, PCR-based molecular markers required only a small quantity of DNA to start with in comparison with the markers based on hybridization. In the additional materials of this subunit, you will find a detailed video about the PCR. Let's see the RAPID. Rapid Random Amplified Polymorphism DNA was developed by Welsh and McClelland in 1991, and it was the first PCR-based marker technique, and it is by far the simplest. Rapids are DNA fragments amplified by the PCR using short synthetic primers, generally 10 base pair of random sequence. The method is based on enzymatic amplification of target or random DNA segments with arbitrary primers. In this reaction, a primer pair anneals to the genomic DNA at two different sites on complementary strands of DNA template. If these priming sites are within an amplified range of each other, a discrete DNA product is formed through thermocyclic amplification. On an average, each primer pair amplifies several discrete loci in the genome, making the assay useful for efficient screening of nucleotide sequence polymorphism between individuals. Amplified products, usually within the 0.5 and 5 kilobases size range, are separated by agarose gels in the presence of ethidium bromide and revealed under ultraviolet light, and the presence and absence of band will be observed. So the advantages of rapids are the low quantity of DNA template required, the easy and quick of the assay, no need prior information for primer design, and the abundance of the random distribution of them throughout the genome. On the other hand, the disadvantages of rapids are the low reproducibility, the requirement of a high-quality DNA in order to avoid contamination, because the short random primers are able to amplify fragments in a variety of organisms. Also, the inherited problems of reproducibility make rapids unsuitable markers for transparency or comparison of results among research teams working in a similar species and subject. And also, rapids are not locus specific, so band profile cannot be interpreted by terms of loci and alleles, so dominance of marker, and similar size fragment may not be homologous. So let's move to FLPs. FLPs, amplified fragment length polymorphism, developed by Keijin in 1990, is a technique based on the detection of genomic restriction fragment by PCR amplification. It's a combination of FLPs and PCR technology. The fingerprints are produced without any prior knowledge or sequence, using a limited set of genetic primers. FLP procedure mainly involves three steps. First one, restriction of DNA using a rare cutting and commonly cutting restriction enzyme simultaneously, followed by ligation of oligonucleotides adapter of defined sequence, including the respective restriction enzyme sites. Selective amplification of sets of restriction fragment using specifically designed primers To achieve this, the 5' region of the primer is made such that this would contain both the restriction enzyme site on either side of the fragment complementary to the respective adapter, while the 3' ends fragment for a few arbitrarily chosen nucleotides into the restriction fragment. And third, finally, gel analysis of the amplified fragments. Advantages of AFLPs. The AFLPs analysis depicts unique fingerprints regardless of the origin and the complexity of the genome. In fact, most AFLPs fragments correspond to unique positions on the genome, and hence can be exploited as landmarks in genetic and physical mapping. So AFLPs are extremely useful and as tools for DNA fingerprinting and also for cloning and mapping of a variety specific genomic DNA sequence. Also, no need for prior information. Compared to other markers, are quite fast to assess. Also, they are relatively inexpensive. They provide highly polymorphism since it can be used with different selective enzymes and primers, and finally are highly reproducible. The disadvantages of AFLPs. They are dominant, so the presence of a band could mean the individual could be either homozygous and heterozygous, low content of genetic information per locus, null alleles are not detected, and are partly laborious. Until now we have seen dominant markers. For now we are going to review codominant markers. One of them is CAPS, cleaved amplified polymorphism sequence, which is an extension to the restriction fragment length polymorphism, the RAPDs method, using polymerase chain reaction, the PCR, to more quickly analyze the results. Like RAPDs, CAPS works on the principle that genetic differences between individuals can create or abolish restriction endonuclease restriction sites, and that these differences can be detected in the resulting DNA fragment length after digestion. In the CAPS methods, PCR amplification is directly across the altered restriction sites and the product digested with the restriction enzymes. When fractionated by agarose or acrylamide gel electrophoresis, the digested PCR product will give readily distinguishable patterns of bands. As I said before, CAPS are codominant markers and they are highly reproducible. The main difficulty is prior knowledge of the sequence of the DNA fragment that is required, in addition to the high price of some restriction enzymes. Another codominant marker is SCAR, sequence characterized amplified region, that takes advance of the polymorphism identified by RAPDs or other markers. When a potentially interesting band is identified on the gel, the band is cut and removed from it. The DNA fragment is cloned into a vector and its sequence is obtained. Then specific primers are designed from 16 to 24 based in length, and the locus amplified by PCR. SCARs have a simpler pattern than RAPDs since they amplify just one locus. However, they required a minimum knowledge of the sequence, and the degree of polymorphism obtained is quite low. So let's move to a very important molecular marker, the SSR, simple sequence repeat, or microsatellites. Is a repetitive DNA in which certain DNA motifs ranging in length from 1 to 6 or more base pairs are repeated, typically from 5 to 50 times. Microsatellites occur at thousands of locations within an organism's genome and have a higher mutation rate than other areas of DNA, leading to high genetic diversity, especially suited to distinguish closely related genotypes. The advantages of SSR are they are codominant, low DNA quantity is required, they have high polymorphism, the genotyping throughput is quite high, Most SSRs are codominant and locus specific. Startup costs are low for manual assay method once the markers are developed, and the SSR markers are highly reproducible and robust. On the other hand, the disadvantages of SSR are the development their development is labor intensive. SSR markers development costs are very high. Startup costs are high for automated SSR assay methods. Developing PCR multiplexes is quite difficult, and some markers could not be multiplexed. So, to resume, the most important messages of this subunit are: first, PCR-based molecular markers are more efficient and less laborious than those based on hybridization. Codominant markers are more informative than dominant markers. And SSR are very good markers since they are abundant, highly reproducible, and robust.